Dear students, in the last lecture we had studied the pathogenesis and the defense mechanism offered by our body in response to bacterial infection, and we had studied some host parasite relationship. The first was benign parasite, which involves the parasite not causing any symptoms or disease in the body, whereas inept parasite overpowers the body's immune system and develops the diseases. Opportunist parasites were present as normal microflora, but in certain conditions they causes diseases, as in case of Diplococcus pneumoniae, which is present in the pharyngeal region. In the pathogenesis, we had studied the exotoxins and endotoxins, which are produced by the bacteria. Then we had studied the host defense mechanism, and we had divided the host defense mechanism into two parts: the non-specific defense, that means the defense mechanism which was not targeted against any particular kind of antigen, and these involves mechanical defense, the cellular defense offered by many macrophages like Kupffer cells in liver, the lysozyme present in saliva, tears. Then we had physiological defense mechanism also in place in response to the infection. Inflammatory defense involves production of inflammatory substances which causes vasodilation and thereby affecting the diapedesis of neutrophils and monocytes, which are phagocytic cells or the of WBC kind. Then we had studied that fever is also helpful in counteracting the effect of infection. The specific defense mechanism involves the production of antibodies against a particular type of antigen, which involves naturally acquired immunity when a person is exposed to antigen in daily life, naturally acquired passive immunity, which involves transfer of antibodies from mother to infants, artificially acquired active immunity by vaccination, and artificially acquired passive immunity by injection of the antibodies itself, as we do in case of snake bite, and also we are doing in case of coronavirus when we are introducing the antibodies in the patient suffering from coronavirus. As far as humoral immune system is concerned, it is constituted by B cells. These develop various antibodies in response to a particular kind of antigen. Heptans are low molecular weight antigens which are non-antigenic unless they are attached with any protein or carrier molecule. They becomes antigenic whenever they are associated with a carrier molecules, and at times they are used in production of monoclonal antibodies. Then there is another system in place for defense, which is cell mediated immune system. This is formed by T cells, which are none other than the lymphocytes which are formed in bone marrow, but they get matured in the thymus. These cells, they generally act on the intracellular parasitic infection. Whenever virus or any other intracellular parasite they enter into the body, they by certain mechanism they kill the entire cell, thereby relieving the body from infected cells which have become useless. Then in epidemiology we use many terms which we ought to know to understand the mechanism of infection and epidemiology. Parasite, as you all know, when an organism lives at the expense of host or derive nutrients for its growth at the expense of host. Infection is actually the growth and multiplication and establishment of disease in the host. The pathogens are those organisms which causes disease. uh, Pathogenicity is the ability of pathogen to cause a disease. Non-pathogenic genicity is inability to cause disease. Pathogenesis is series of events properly establishing a pathogen in host and development of disease. This is very important. Let me repeat it again. The series of events which establishes a pathogen in host bodies, and thereby we develop a disease. This mechanism is called as pathogenesis. Epidemiology has its etymology in epi means upon, and demo means population, and logos means to study. The science which deals with occurrence, determination, uh, distribution and control of disease is epidemiology. Let me repeat again. Occurrence, determination and distribution and control of disease is epidemiology. What are endemic diseases? A disease which maintains a steady low level frequency at regular intervals, like common cold. They are called as endemic diseases, as divorced from pandemic diseases which manifest at wider areas. Then epidemic diseases, a sudden occurrence of disease beyond the limit is called as epidemic disease. It remains largely uncontrollable and counteracted by taking certain measures and vaccination. Pandemic disease, pan means all. When a disease occurs in a large population over a wide region, it is called as pandemic. Like in case of coronavirus, we are just undergoing pandemic type of uh, viral spread. Prodromal stage is the period before the outset of the or the commencement of the symptoms of a disease. Incubation period is the time period between infection and appearance of disease. What is convalescence? It is the period of decline when signs and symptoms of disease disappears and recovery is effected in the body. Herd immunity, these days you are hearing it widely. Development of immunity in large percentage of a population so that infection and spread of pathogen diminishes. Public health officers make sure that 70% of the individuals must be immunized to maintain the herd immunity. That's why a vaccine is much desired in case of corona pandemic, because we need to immunize 70% of the total population of the world or the country to counter the effect of corona pandemic. Antigenic shift. Some pathogens continuously changes their antigenic determinants. That means, therefore, they could not be recognized by our immune system because of the small changes they make in their antigenic determinants. This may result by hybridization between two antigenic types, like we are having many types of AIDS virus, influenza virus, and we are many strains circulating as far as coronavirus is concerned. Antigenic drift. Smaller antigenic changes in pathogen to escape the immune system of the host is called as antigenic drift. This is very similar to antigenic shift. Then we have antimicrobial chemotherapy. Whenever we encounter a pathogen in our body, we just support our immune system with some chemotherapies and other mechanisms. Chemotherapy was first of all developed by Paul Ehrlich. Sulfa drugs invented by Dogmack. Penicillin was discovered by Flemings. and streptomycin was invented by Waxman. Now, what are the general features of these antibiotics? Penicillin acts against gram-positive bacteria. I told you when I taught you uh, the basics of the microbiology that when I taught you difference between gram-positive and gram-negative bacteria, that penicillin acts against the gram-positive bacteria like streptococci. Streptomycin generally acts against gram-negative bacteria like Escherichia coli. Narrow-spectrum drugs are those which are effective against limited number of parasites, whereas broad-spectrum drugs or antibiotics, they are effective against large number of parasites. Antibiotics are secondary metabolites and their potency is expressed in units per ml. Now, what is the mechanism of action of these antibiotics? They affect the survival of the pathogen in various ways. Like penicillin, it inhibits the cell wall synthesis in bacteria. Penicillin inhibits the transpeptidation enzyme responsible for cross-linking of polypeptide chain of peptidoglycan and also activates the wall-lytic enzymes. Penicillin is bacteriostatic. Mesosome stops wall synthesis, which is prerequisite for the bacterial growth and division when we supplement the body with penicillin. Polymyxin B, it disrupts the bacterial cell membrane, and gramicidin alters the membrane nature, thereby rendering the parasite ineffective. Inhibition of protein synthesis is done by streptomycin, which binds to 30 Sydberg unit ribosomal subunit and misleads the messenger RNA chloroamphenicol, and E-mycin binds to 50 Sydberg unit or the largest subunit of the bacterial ribosomal subunit, inhibits the peptide chain formation and elongation by inhibiting the peptidyl transferase. You might have studied peptidyl transferase when you have studied that translation mechanism. Rifampin, it inhibits the synthesis of purine and pyrimidine, thereby inhibition of DNA synthesis is effected. Mitamycin blocks the synthesis of DNA. Then respiration of bacteria inhibited by gramicidin and valinomycin. These inhibits the oxidative phosphorylation in bacteria. Sulfonamides, they are antagonists to the metabolic pathways of the pathogens. Now, one term you need to remember is drug resistance. When pathogen checks the entry of the drug inside the cell, they become resistant to drug. Like penicillin is not allowed to penetrate by gram-negative bacteria. This is a kind of drug resistance developed by gram-negative bacteria. Secondly, bacteria pump the drug outside the cell with help of an enzyme called as translocases, which is a transport protein. Even if we are able to introduce the antibiotic inside the bacterial cell, there are certain proteins which can pump it out back to the exterior, thereby saving the pathogen from the destructive effect of the drug. The various transport pumps are called as multi-drug resistant pump, which have been developed in the bacteria lately due to overuse of the antibiotics. Thirdly, we have modification of drug itself by bacteria. because the bacteria, they have developed the capacity to modify the drug. Like penicillin is hydrolyzed by penicillinase and making it or turning it ineffective. So students, this is all in this lecture. Thank you very much.